Welcome everybody. I'm David Hart. I'm the director of the Mitchell Center. I'm going to try to get this going quickly because it's a kind of a 1, 2, 3, 4, 5, 6, something like 6-ring circus. But I just want to say for a second, if you've seen some of this stuff go by, a few of these are related to talks that people in this group have given or presentations in various places. I also want to mention that this Thursday, coming up is Ruth DeFries from Columbia University giving the Mitchell Lecture on Sustainability. And if you're, there's still tickets, but you do need tickets. They're free, but we just try to keep track and make sure we don't get into space problems. So please check with Ruth Holsworth or Carol Hamill or me if you need tickets. And if you are interested in meeting with Ruth while she's here, I'm not saying there won't be a lot of things going on on Thursday, but we'd be glad to try to make that work for you. So if you haven't figured out a way to do that yet, let us know. So I just want to say that everybody that's presenting today, it's been my pleasure to work with as folks that have made a big difference here at the Mitchell Center. Some have, many of them have strong connections, and I'll just do a quick introduction of the folks and say something about that. So Bridie McGreavy is going to lead off. She's an assistant professor in the Department of Communication and Journalism, but she also got her PhD and work connected with the Mitchell Center, did a postdoc on this project in connection with the Mitchell Center, and now she's co-PI on our new NEST project on the future of dams. Brian McGill is in the School of Biology and Ecology and also has a joint appointment in the Mitchell Center. Sean Smith in the School of Earth and Climate Sciences, also in the Mitchell Center. Sam Roy, who did his PhD in the School of Earth and Climate Sciences, then was a research associate on the Safe Beaches and Shellfish project, and partly because he found that quite enjoyable and we appreciated what his skill set was and his unique abilities here. He's now a postdoc on the Future of Dams project. Kate Beard Tisdale is a professor of spatial informatics in the School of Computing and Information Science, and was in, I forget, up on the very first day of SSI, but early on, way back, probably in 2008 or 9, if not before. And Damian Brady, it's been a pleasure to work with him. From the Darling Marine Center, he played big roles, as you'll hear, in this particular project. And I'll just say that I think there's huge opportunities for us to be doing more and more work on decision support. And so this is one of many efforts that are going on either in conjunction with Mitchell Center or in other places on campus. So I think it's worth us thinking about what are we learning from this effort compared to others, and where do we go from here. But to me, the future is very bright. So, Bridie, take it away.
Thanks. Thanks for coming. We're really happy to be able to share this work focused on creating a decision support toolbox for safe beaches and shellfish harvest, which reflects about 3 years of focused effort. And thank you, David, for that introduction. I also wanted to acknowledge other people who have been centrally involved in moving this work forward in a variety of ways, but who are not co-presenting today. So David Hart and Kathleen Bell are co-PIs on the Safe Beaches and Shellfish Project and the NEST New England Sustainability Consortium. Kelly Cole, who is a postdoc working with Damian Brady. Frank Hsu, PhD student working with Kate. Benoit Parmentier, postdoc working with Brian McGill. Brett Gerard, who's here, PhD student working with Sean. And we had involvement from multiple stakeholder partners, which was very valuable, but I also wanted to acknowledge the really important role that Cole Canwit, who is the director of the Bureau of Public Health at the Department of Marine Resources, and Carrie Kazor, who is a coordinator with the Maine Healthy Beaches. Both of them played really important roles throughout the project and to today. So, as David mentioned, this effort was an outgrowth of the Sustainability Solutions Initiative, or SSI, which launched the Mitchell Center for Sustainability Solutions. We received a $6 million grant awarded to the University of Maine and the University of New Hampshire to launch the New England Sustainability Consortium. NEST is our acronym that we'll be using, and create this first project in the consortium, the Safe Beaches and Shellfish Project. It primarily involved a collaboration between these two host institutions, as well as many other academic, governmental, and non-governmental partners, many of whom you see in the logos listed here. So DMR, Maine Healthy Beaches, College of the Atlantic, Frenchman Bay Partners, and Maine and New Hampshire Sea Grant, among others. Our main objective is to find multiple ways of linking science and different types of knowledge with decision making. So this project focused on the decision support toolbox was one of a host of things that we did in Nest and on Safe Beaches and Shellfish to do that. We're going to be taking a tag team or relay approach to our presentation. A lot of people involved, a lot of different components. So we wanted to provide two organizing slides to help you keep track of how these pieces fit together and how we organized ourselves in our work. First and foremost, we used a sustainability science approach where we had active and iterative stakeholder engagement in all phases. You know, many of you have been using sustainability science in different ways, but I will provide a brief overview of some of the principles of sustainability science, recognizing that there are students who are new to this type of work, so I'll say a little bit about that. If you've ever worked in any kind of partnership, and certainly interdisciplinary partnerships, you can recognize some of the challenges inherent in bringing different ways of understanding the world, different research traditions, different methods together. So in a project or on our team that included ecologists, fluvial geomorphologists, information scientists, communication researchers, marine biologists, and others, we ran into some of those same challenges too. So we used an approach that has been very influential in sustainability science that's known as boundary work and the use of boundary objects. So these are things like technical reports or conceptual frameworks. If you pulled a map out at a meeting, you know how maps, people just glom onto them, and it can help in translating the differences and coordinating across difference, negotiating, sometimes reinstating the difference. So they're entities, these tangible things, but they also set up these processes, these open-ended creative processes. So we early on, recognizing some of the challenges in bringing these different ways of understanding the interactions at the land-sea interface together, developed this sources delivery residence time as a boundary object, and Sean Smith deserves a lot of credit for that. So he'll be talking about how that has helped us bring our differences together and work together. And then you'll also see two scales of analysis that, again, reflect what we're hearing from stakeholders in terms of needs for information, and then also what we needed to do to actually produce science on this project. So we're looking across the coast in Maine and New Hampshire. We'll be focusing more primarily on Maine today. And then at reference watersheds in Bar Harbor and Wells, but we've also in Safe Beaches and Shellfish done work in York, the Madomic, and Machias as well. We'll also be talking about the different analytical approaches that compose the decision support system toolbox, including spatial data assembly and analysis. In the first year of the project, we basically got this massive data set from DMR that included water quality information and rainfall information. And DMR is using this data set on a day-to-day basis to make decisions about closures, but they asked us for help in trying to understand what's really going on and how could we use this information to fine-tune our management. So as the team stepped into doing that kind of work, they recognized that there were some major data gaps and started to fill those gaps, so they'll talk about that, and develop an enriched spatial informatics and statistical approach to understanding this relationship between watersheds and estuary processes and water quality. And that also involved hydrological modeling and circulation modeling. In each of the presentations, we'll be addressing how do we connect with stakeholder questions, what were the key results, and what are the next steps. Because one of our major insights is that 3 years goes like that, especially with the level of complexity in the system. So this is still a work in progress, but we've certainly made a lot of progress. In terms of sustainability science, we commit in this work to addressing complex problems, and this has two parts. In terms of addressing the problems, we want to understand these problems from multiple perspectives. And then we also want to do something about it. We want to try and advance different types of solutions to these problems that other people have referred to them as really wicked, where you think you're fixing one part, you end up breaking another part. There are cross-scale interactions, unknown thresholds. The problems keep on changing as time goes on. But these are the kinds of problems that we're really interested in. And we approach them by setting up this adaptive, iterative engagement that brings together interdisciplinary perspectives. We want to understand these problems from many different disciplinary standpoints. But we want to work with each other as researchers and with stakeholders to create a pull for our science. And we do that by systematically understanding what are the needs for information. We've also been very informed by Linda Silka's work on community-university partnerships, as well as Eleanor Ostrom, who emphasizes that we need to go beyond panacea thinking. And then Karen Hutchins, who's now BLIC, who I hope is listening in on the phone. She said she was going to. In her work in SSI, working with Linda and others, she looked at partnership preferences. So we want to understand, how do people want to be involved with us? In some cases, you might have stakeholders who say, here are my needs for information. Here are the questions that I have. You go off and do the science, and then at the end, we'll be ready to use it. In our case, because one of our primary stakeholder partners was the DMR, they needed to be involved much more fully, especially in the later stages of the project, because they then needed to be able to describe and justify the science to the Food and Drug Administration, the Interstate Shellfish and Sanitation Commission, these other regulatory agencies that guide their management. So we were adaptive in how we were working with these partners to help them be involved in ways that they needed to be involved. And we think that this has been fundamental in our ability to connect science with decision making. So just a quick snapshot of some of the complexity of these problems. Be focusing just in the interest of time on issues in the shell fishery, but certainly our work focused on beaches as well, and there are similarly vexing problems there. Shell fishing comprises the second largest fishery in Maine, about 22 million in landings value in 2015, 2,200 harvesters, commercial harvesters, who rely at least in part on this resource. It's also a really interesting example of co-management and local governance. So you have towns that are responsible for stewarding the resource, for doing conservation activities and those kinds of things, working with DMR, whose mission is to protect public health. This is my bias as somebody who has a clam license, but I also think it's a really delicious and healthy local source of food, if you've ever had fresh clams or mussels. And given that this has been a source of sustenance and in later years trade for somewhere around 9,000 years on the coast of Maine, it has rich culture and traditions and meaning for coastal communities. I do a lot of work with clam diggers, working with Tyler Quiring and others, and I'm regularly amazed at the kind of stories that they tell about things that they notice out on the flats, how they're noticing changes they're going on, how they're responding. And then in their involvement in municipal management, it's in their best interest to get involved in this and manage the fishery because it's in their economic interest. But there are so many guys, and I say guys because it's primarily a male industry, who go way above and beyond what's needed. And the kind of volunteerism is really quite astounding. So managing this system from a public health standpoint is complicated. This is DMR's primary mission. And they look at approximately 215,000 acres of shellfish growing area that come under technically 4 different pollution classifications, but here we have prohibited or restricted. There are about 186,000 acres that currently have those designations. These are acres that have long-term closures. And then there's about 28,000 acres that are subject to these shorter-term closures under adverse events. So when you have something like 2 inches of rain in a 24-hour period, the flats can be closed for up to 2 weeks. And there are other events that can shut them down as well. But this is a major economic impact for harvesters, and it really increases the uncertainty in their lives. So in our interest in doing something, linking science with decision making in this context, we set up a stakeholder engagement process that had key phases of information gathering. We needed to learn as much about what was going on in the system, what are the problems, what are the needs for information. Started our project in September 2013, spent the first several months producing with stakeholders a technical report that helped us understand some of the issues in the shell fishery and in beaches, and also helped us identify some key stakeholder partners. We conducted about 40 interviews over the course of a year where we, again, scoped out information needs and partnership preferences. We started to synthesize this information and feed it back to our stakeholders. The Shellfish Advisory Council, Shellfish Focus Day, Maine Water and Sustainability Conference, we started to hear from them. We need more involvement. We really want to sit down with you and unpack what's actually going on. And so we started to set up more co-production meetings where it was much more focused on dialogue. We heard a lot of different types of questions, but we grouped these into two broad types: management questions. For example, does it make sense to have a one-size-fits-all trigger? What are the data gaps? And then context questions that spoke to stakeholder needs, but also to our needs as a team to produce science. So how does context matter? And what are the explanations for problems and variations in response across the coast? And then ultimately, how do we develop better tools for explanation and prediction? So these are questions that the team will address, and Brian McGill is up first.
So I'm been working at the whole coast scale, all of Maine. Been working closely with Kate, with Benoit Parmentier. And I'm going to take the risk and try and see if we can do the live demos. This is a website, and as was mentioned, broadly working in the context of decision support systems, which to me is bringing data and models to end users to inform decisions. And it can come, having said that, that covers quite a large span of territory, ranging from just very simply bringing data to the end user through a simple web interface like I'm about to show here, or it can be bringing more derived data through the very complicated simulations and analyses, which I'm going to give an example of in a minute. So kind of both ends of that decision support spectrum. So one of the big issues is there's a lot of data collected at sites, in these cases at the Shellfish Flats. And they collect a number of data. The target data is bacterial loads, but they are also looking at, they measure salinity and precipitation and temperature and some other things like that. But one of the key variables that they focus on is rainfall. And they have a variable that records what the rain looks like at the moment that they're out there taking the bacterial sample, but it's missing a lot of the rainfall context, which is spatially larger, right? A whole watershed is feeding in, and it's also temporarily larger than the hour that they happen to be there. So we were able to merge. One of the big things we did, my team of the 5 teams here, is to merge third-party rainfall data with the DMR data so that those two things can be put together. And then taking that rainfall data and their existing data and just putting it into a simple web application. So I'm not going to spend a lot of time on this, but you can change the dates that you're covered. You can change the station. And this is just a simple example of the data. So the black line is the rainfall, in this case through the year 2012 at a particular station. And these red dots, red crosses, are the actual measurements. So the places that you don't see red are places they didn't have measurements. It's a not exhaustive sampling system. They sample periodically. And you can see that there's some clear links. Here's a day with a pretty heavy rainfall and a day with a pretty heavy bacterial load. Looks like the same thing's happening here. Some of the other days they came to visit without a bacterial load. There wasn't rainfall. But this is just directly accessing the data that they have, and we can switch stations, for example. So this lets them actually get into their database. Previously, you pretty much had to know SQL, a technical language, to get into their database. And I'm not going to run it because time is pretty tight, but this is a map of rainfall and bacterial loads, and you can zoom in on that and see how those are changing through time with a spatial context. So I'm going to switch back to the presentation. So that's a very simple tool, very simple decision support. It's their own data, but it's putting it into mixed with the rainfall data and putting it into an access context that is quite a bit simpler. And you don't have to be a technical expert to be able to go look at the data. The other context, the other end of DSS, is doing sophisticated analyses. And in this particular, I realized right before the presentation, I duplicated one slide and didn't get the other slide. But if you think about going back to this kind of data, you can actually study the relationship with a regression between rainfall and the bacterial loads. And so you can just do a regression. We also brought in other data, salinity, temperature, and time of the year, which is hypothesized to matter. And there's over 2,000 stations. We ran a regression for each one of these stations. And the net is rainfall matters, salinity matters, temperature matters, time of year matters, interaction between precipitation and time of year. All of those things matter. So a rainfall in July matters more than a rainfall in May. All of those things matter. We explain about 30–40% of the variance. Most of the regressions are significant. And most of these things work in the direction you would think. Higher salinity is lower bacteria. Higher temperature is higher bacteria. Later in the year is higher bacteria. The rainfall one was kind of interesting. There were a lot of significant relationships with rainfall, but it would very much vary from station to station. And actually about half the stations had a negative relationship, where rainfall was lower bacteria, and about half the stations had a positive relationship with higher rainfall, meaning higher bacteria. So if homing in on the relationship between rainfall and bacteria, this is a color-coded map. You'll recognize Penobscot Bay and down towards Camden, Rockport, etc. This is what we would call a sensitivity. This is the slope of the relationship between rainfall and bacteria. So if you see something that's on the yellow end, that means when the rainfall is higher, the bacterial loads on average are higher. And when you see something on the blue end, that actually is a negative relationship. And it's hard to do statistical analysis on a jagged coastline like this, but you can certainly start to see the islands tend to have a lot of positive sensitivities. Rainfall is directly affecting the bacterial loads on the islands. It's a little bit of an upstream effect too, where the rainfall is having a bigger effect upstream and down towards the mouth of the bay onto the ocean. And the last thing we did was trying to really home in on this 2-inch rule. The data is sparse, so it takes some tricks to analyze the data, but here's a couple different contexts. This is asking, after a 1-inch rainfall event, how many times higher is the bacterial load than on a day when there wasn't rain? And so in most cases, it's 1 to 5 times higher after a 1-inch event than on a day when there wasn't rain. Similar stories for a 1.5 or a 2-inch event, although you can see the 2-inch event is definitely skewing things to the right. Or we can flip it around and say, if there's a threshold in bacterial load, I took 50, but 30 is another common threshold. It doesn't actually change the story that much. What was the rainfall on the day in which we had bacterial thresholds exceeding the 50% level? And a lot of times they exceed even when there was not a rainfall. But you can see that clearly fairly heavy, these are quite heavy, unusual rainfall events are common on the days when the bacterial loads are high by some definition. So just kind of wrapping up, we had that very simple DSS that's just bringing data to stakeholders, and then we have a much more complicated analysis. This represents thousands of regressions and lots of different ways to look at the data, but it's also summarizing the data in a way that can be looked at. And in particular, we can look at the spatial context and the seasonal context, and both of those, as well as the salinity and temperature context. And all of those things are modifying that 2-inch rule. The bottom line is there's an enormous amount of variability and an enormous amount of complexity, but there are also clearly some patterns in here that can help towards management decisions.
Okay, so I'm going to cover some of the work from the Watershed Process and Sustainability Research Group, which is centered in the School of Earth and Climate Sciences, but interacts with the other participants in the project, including folks on Mount Desert Island, the MDIBL lab, and University of New Hampshire, and of course with Bridie, with our interactions with the stakeholders along the way. And key participants in this effort have been Sam Roy, standing next to me, Brett Gerard, myself, and Abby Bradford, who was a summer student and then became a graduated staff employee of the research group and did a lot of work on the GIS. The map there that you see is the coast of Maine, of course, and throughout its extent, and you see these red lines or polygons in there. So a lot of our attention is on the runoff relations. And so I have here, we were charged with talking about this toolbox, and I think it's a little bit loose to frame things around a toolbox because there are different perceptions of what that means. So in the top, I have an empty toolbox that says that maybe you'd have some fundamental understanding of processes. There are textbooks about watersheds and how they route runoff and pollutants down from the top to the bottom. There are also textbooks about estuary dynamics, but if there's limited knowledge of a coastal system, which is our domain, then there's a difficulty in actually making something that's predictive and useful for a manager. In the lower two clusters of words there, the some knowledge of the domain but limited quantification of land-sea connections can actually give you a partial toolbox where you have some things to actually do make some predictions, and in this case it may be a universal 2 inches of rainfall closure criteria, but there are a lot of there's a lot of variability, as Brian was mentioning. Pollution events have been observed on the coast. There's coincidence between rainfall and pollution, and that's been pretty well documented by sampling that's occurred over the last decade or more. But there are knowledge gaps. There's relations between rainfall and runoff, and that's the amount of precipitation that's converted into something moving across the surface and carrying constituents with it. It might be sediment, it might be nutrients, but it might be bacteria. The relations between runoff and pollution as a result of that, and there's identification of specific culprits that needs to be done in varied settings. Watersheds are not created equally. They vary based on what the past history has been, including the glacial history, but also the history of since European colonization of the landscape. So a toolbox assembled from scientific research might address some research questions that are focused, and it might have information that's assembled and through literature review and the development of spatial datasets and other datasets related to precipitation, water flow, concentrations, and they usually produce results at a site or at a site scale or specific sites and at a scale that's specific to the research questions at hand. The task that we had was to put together this toolbox that had management purposes, and the way that that's out a little bit differently is that its organization is done by process categories that are relevant to runoff and pollution transmission into coastal areas, evaluated over multiple scales. There's attention to transferability or generalizable results, explanations for the approach, and adaptability. And to do this, we focused on 2 different scales. One is the Gulf of Maine coast scale, which I showed in the first cover title plot, but the other is this boundary object that also references this scale, which is was mentioned by Brady, that divides the world into source, delivery, and residence time culprits, and has that co-production space related to communication of these different culprits involved with coastal pollution. Pinning these down at the scale of the Gulf of Maine is a very large task, and there are a lot of proxies that are necessary to do this work. So we also work at a second scale, which is the reference watershed scale, and we're working in near Bar Harbor at Cromwell Brook and the Wells Estuarine Research Reserve to pin down these processes and to explain them in more detail to defend our use of the proxies at a larger scale and a looser manner. So at the Gulf of Maine scale, the watershed metrics that were selected are related to whether these different watersheds have source differences related to runoff production, for instance, soils and impervious area that creates rainfall runoff in varied amounts, or sources of bacteria pollution. Humans, of course, are a source. Wildlife can be a source. And Steve Jones at the University of New Hampshire has found a number of other types of sources in these systems. But the sources are produced in the landscape, and not all of them reach the coastal estuary. The delivery is also varied. They reach the estuaries by rivers and overland flow, and the mechanics of those vary based on the history and the current conditions. And then finally, the runoff is placed in a tidal estuary and has varied residence times. So these were all the culprits that we had to assemble for the coast using proxies and available spatial data. And I can tell you that one of the advancements of this project is that it took a lot of work in order to get the spatial data arranged and framed so that we could actually start addressing these questions with reasonable accuracy. And one of the first steps was just to get a data set that had all those polygons represented in an accurate way using high resolution elevation data. So once we were able to parse these watersheds into their drainage divides, the next assembly was to take those source culprits and delivery culprits and compare watersheds through a principal components analysis, resulting in 6 different types of watersheds that we identified on the coast, which breaks down the problem rather than 500 watersheds into something that's more manageable on a scale of 6. And you can see these differences that are fairly general. Some of the watersheds are characterized by high relief, wide estuaries, and thin soils. Others have high storage, high agricultural use. We have one for high average conditions, urban conditions, which of course would be areas like Portland and some other areas of concentrated urban growth. and history, and buildings and impervious area, everything that goes along with it. And then there's low relief bedrock drainage controls. Once we have those broken out by those parts, we have 6 clusters on the coast. We can relate those to the history of bacteria exceedance values off the coast, which is shown a little bit less clearly here, but you see all these red dots along here showing these different types of bacteria scores. Then we have the ability to compare, often the plot on the right-hand side of your view screen. The clusters are in the x-axis there, C1 through C5, and you see the coliform most probable number per 100 milliliters, and you see the statistical differences in those that are significant. We had some trouble with this. We actually used the 90th percentile value of the cumulative distribution of these bacterial exceedance values and found some similarities between the basins, but we also now see there are differences. At the reference site scale, these are some of the variations that show up from the top to the bottom. We have a drainage network coming from Cadillac Mountain down into Bar Harbor. You can see how some of this variability shows up in the landscape. We're looking at a distributed watershed model in those areas to explain the mechanics. So the residence time deserves more treatment than the other parts. It's a bit more complicated, and I'm going to let Sam go through 2 of the slides.
All right, thanks, Sean. Okay, so I'll bring it back to the Department of Marine Resources management question. Does it make sense to have a one-size-fits-all closure trigger? So as Sean mentioned from the box and blister plot previously, all watersheds do have some contamination risk. But it could be that for given watershed to watershed, the time interval and the spatial scale of that risk may differ significantly. Okay, so that could mean some important implications for this one-size-fits-all. 2 inches of rainfall within 24 hours equals 2 weeks of closure for shellfish and flats. So from a hydrologist perspective, how do we go about exploring this question? So what I've done is we've just gathered a significant amount of geospatial data, which could be used to, along with combining it with simplified runoff model and an estuary mixing model to cover a lot of ground in a short amount of time. We can make estimates of how much runoff is being brought to the estuary, which is significant in the transport factor for coliform bacteria, which is coming from the surface, but also for its survivability within the estuary. It requires, or most of them require, fresh water to survive. How long does it take to get there? So we can use just pretty simplified hydrologic models to figure out how much time it takes for a parcel of water within your watershed to transport down a river to the outlet at the sea. That's based off of geospatial data for drainage area, for surface roughness and channel slope. And finally, how long does it take to mix or remove that runoff when it gets to the estuary? So in order to answer that, or at least explore that component, we're using a simplified tidal mixing model, which uses bathymetric data and tidal range data to figure out the capacity of mixing and dilution for 535 estuaries along the coast of Maine. So an estuary or coastal landform feature for all of these watersheds. So again, these are pretty lightweight models. We're trying to just get some idea of the mixing capacity, residence time for all these watersheds. And with that, we could maybe pinpoint reference watersheds that we could study in more detail to get more information from. Okay, so I forgot to mention that I'm looking specifically at the St. George in this example. The reason why is it's a pretty high productivity site for shell fishermen, and it's also considered to be high risk with DMR. So looking at the St. George estuary, this blue region is the estuary, and the limit here is actually shows you the spatial limit of a high concentration of runoff after a 2-inch 24-hour rainfall event. And within this region, the maximum residence time calculated from that mixing model is 4.5 days. So we've done this for all of the watersheds across the coast. Some of the results, runoff mixes sufficiently before 2 weeks for many of the estuaries, which is a pretty interesting, surprising result. That could mean some major implications for this automatic 2-week closure. Of course, there's so much more work that needs to be done before any changes take place. But it could mean that the one size fits all may not exactly be important for some of the watersheds. And of course, your concentration and the residence time depends on where you are within the estuary, the timing and intensity of the rainfall events, and the concentration of your contaminants, which are coming from the coast, from the land, which could be tied to point sources, non-point sources, or stochastic events such as pipe breakage or spills or something like that. So critical further work that we need to pursue with this to just to sort of solidify our trust in this model is to calibrate further this data with some of the salinity data and coliform scores. We started doing this, but more work is necessary to really get a good calibration for this model. We need to include nearshore sources, what I think Kate might start talking about today later on, and we need to coordinate with estuary scale circulation models, which is what Damian will be working on.
Great. So at this point, we've summarized the source, delivery, and residence time and approaches for the analysis of those components. So I've divided things up here into the toolbox and workbench outcomes. The toolbox, we have new customized spatial data sets that not only feeds into our conclusions that we have now on spatial variability and runoff and the movement of that runoff through estuaries. We have coastal landscape setting IDs based on those three culprits that we've identified that are relevant to pollution management on the coast. We have watershed vulnerability comparison, and we have estuary vulnerability estimates from that. For the workbench, we have more work to do on culprit dominance by setting. That is sorting out which places have higher sources but low delivery, and which places might have high delivery even though they have a low source, and then the residence time is the other part of that problem and the relevance relative to the watershed dynamics. We have a mixing model validation. Steady, our approach for this Gulf of Maine scale is steady state. We're putting alongside an unsteady state, and that type of a framework for analysis will actually help us confirm our findings and defend them or validate them. Stochastic process evaluation. So some of the correlation that we see makes sense that the urban areas may have a higher association with bacterial exceedance events. But a lot of the processes are going down that are happening along the coast, broken pipes, tipped over trucks, different sources that pop up are random and over long periods of time. We have a watershed feature sensitivity analysis that we're working on the two reference watersheds with to look at the effects of human infrastructure and climate change on these associations with watershed dynamics with coastal estuary dynamics and pollutant loadings, and then quantifying those loads along the coast. What we got?
And I also just want to say that all of our spatial data is readily available as KMZ files. Just send me an email.
So I'm going to cover another aspect, another dimension of this, more focusing on the spatial informatics piece and the statistics. And one of the leading questions that we had is how does context matter? And what are some of the explanations for problems and variations in response? And so specifically, I wanted to look at, in more detail, what are some of the spatial patterns? We're looking at the DMR water quality data. And then how do these associate with some terrestrial influences relating to the three-part, but focusing more specifically on sources and delivery components? And then how do these influences change with scale? And so I'm going to focus on, kind of zoom in on the medomic here and look at it in more detail. And I don't know if this is actually, okay. So what we do see, and this is fairly typical, is that there's higher levels of impairment in the upper reaches of the estuary, and as you move down, it progressively gets a little bit better. But we want to examine that in a little bit more detail. And this is just looking at the geometric means over the data set for a complete forestation, this complete data set. I also, before I forget, I want to thank Frank Zhu and Yuan Wei Wang for helping with the data analysis and compilation. And that's just to situate where we are with the medomic. So a box plot, we saw one in the previous talk. Box plot is a useful informatics tool for looking at, in this case, at the distribution of the log coliform scores for each station, but it's not particularly spatial. So there's a little bit of spatial aspect to this, and that this starts at near the coast, and as we move up, we're up to the head of the estuary and then back down. And what we can see here is that there seems to be a problem as we move up towards the head of the estuary. But it's not really spatial. And so one of the tasks in terms of the toolbox is, can we spatialize this information and get a sense of, again, how are we seeing this variability spatially? So the first step in this was to construct Voronoi polygons for each of the DMR stations. So a Voronoi polygon is simply the area around a point that's closer to it than to any other point. So it's sort of grabbing this area of influence. In this case, it's constrained to be inside the confines of the estuary. So now we have some polygons that we can associate with our observation points. And then the next step was we clustered these stations based on the percentiles of their log coliform scores. So we're taking the minimum, the 25th, the median, the 75th, and the maximum. And this gives us a set of clusters. And they don't come out in the proper order. Number 2, the second cluster is the worst. Again, you see it has the highest mean and the highest 75th percentile and the highest max. Cluster 1 is actually the best. It has the lowest, essentially on all of them. And then 3 is actually 4 is sort of next after 2, and then 3. So this gives us a little idea, again, of how the bacterial levels are behaving spatially within an estuary. And then, as Sam mentioned, Sam and Sean have been working with these larger watersheds and characterizing those. We wanted to look at this in a more detailed spatial context, and so we did some infill with a set of smaller watersheds and characterized them with the same dimensions, looking at sources and delivery. And here we're looking at, so these are all small watersheds, and we're looking at just agricultural, two land cover types, characterizing these watersheds, agricultural and storage. Storage being ponds and wetlands. And some interesting things show up. So we might ask, like, why was this point, and we can say the area around this point, why does it have a particular problem compared to this one upstream, which doesn't? And some of the things that we can see is, well, there's higher agricultural activity here, and then conversely, this is storage. There's less storage here. And if we look at this location here, there's a high amount of storage and really no agricultural activity. And so we see less problems in sort of this part of the estuary. And then just like at this area, again, there's more agricultural influence coming into just this tip of this estuary and less storage. So we start to see, again, context matters, even at a small scale. And just looking at land cover types gives us some indication about what's happening locally. So how do we then connect the terrestrial covariates with and this is one thing that we've been struggling with the points that the DMR have sampled. And again, this is sort of an informatics tool that we considered using linear referencing. And a linear reference is, again, you can think of it as an informatics tool that we create the outline of the estuary, the Potomac in this case, is a linear measure. So it has an origin down here in the lower left, 0, and then it just increments as it wraps around. And so we have a way now of indexing things, features along this linear referencing system, and it gives us, again, a way to link our terrestrial covariates to our observation points, and you could say their areas of influence. So we applied this linear referencing system, and it also, you can create things like parcels as events. So we can intersect the edge of the parcel with, in this case, the Voronoi polygon, and it gives us some summary statistics. So how many parcels are associated with this piece of the coastline, how many inlet streams are associated with that, and any other possible variables that might be covariates that might be of interest in order to link them. And then this gives us, again, a framework for getting a set of covariates to link to observation stations to do, say, regression, cart analysis, and so forth. And then just last, some next steps. So Brian mentioned the PRISM data, and I think this is, we compiled this very large database of independent precipitation data, and being able to link to, we have a long history of DMR data, and you can see in this time series of the DMR, this is the log coliform scores, some exceedance events, and we'd like to explore those in more detail with respect to precipitation events. And so, are there bacterial load responses at locations? Are they similar under similar precipitation conditions? Not just on the day of, as Brian mentioned, but looking at the antecedent pattern of precipitation prior to that exceedance event. So further is linking linked to the runoff modeling piece. Can we use this linear referencing as a tool to help us integrate information across this sort of land estuary divide? And one of the issues is a lot of the GIS database don't give you an easy way to identify a subbasin. If you use linear referencing, you can cut it anywhere and say, okay, I'm going to define my basin from here to here with a linear reference. So you can slice and dice the estuary into chunks and then link it to these various covariates as you wanted to, for your analysis. And then give us an opportunity to link it to these smaller scale watersheds. And these Voronoi polygons are clearly just an approximation of an area of influence, and we would really need the circulation model that Damian's talking about, or will be talking about next, to really get a better understanding of some of these influences within the estuary. And if we just look at, again, context matters, if we just look at this point, I mean, maybe its problem is it's the way it sits with respect to this little point, and maybe it gets trapped in here and the circulation is a problem. So I think this is a case where, again, we start to pull these pieces together, but we may be able to use these informatics tools, the linear referencing piece, as a way to help facilitate, again, the information integration, making it easier also for users to access this information. I'll stop there and turn it over to Damian.
Okay, so if you've learned anything at this point, you should know that estuaries are really annoying. And if we were in any other EPSCoR state, let's say Rhode Island, we would deal with one watershed, the Narragansett, but we have 535, and you'll notice on that map we don't put in the Penobscot or the Kennebec, for instance, which drains half the state. And the Kennebec is draining a third of the state into the same estuarine surface area as the Demerskata. So each of these estuaries is very, very different in their own way. And so figuring out what happens when we've gone from the rainfall data that Brian was showing through the system of watersheds that Sean was showing, and then through Kate's linear referencing system into the water. Okay, so that's where I sort of want to go next. And the transport of this material into estuaries, and here's an example. This is a plume in the Merrimack. This is Dr. Kelly Cole's model. And just demonstrating that once it goes out into the water column, it's really, really difficult to figure out where it's going to go. It's very context dependent. And Brian, I thought, really did a great job of showing that map. There are sometimes positive correlations with bacterial pollution and sometimes negative, and that's due to the nonlinear dynamics associated with transport. Sometimes a rainfall event will flush an estuary, and sometimes a rainfall event just means more loading of bacterial pollution. And that the actual rainfall event also dictates what the transport looks like. So what we wanted to do then was take a reference watershed scale approach. Sean already mentioned this. We looked at wells, and we also did a lot of work in MDI. It's always fascinating to me to visit places like the Wells National Estuarine Research Reserve or Chewonki Campground that's been around for 100 years. Or most people in Maine think of Maine as a very pristine place, and it is, but every single one of them has a pollution closure map that looks like this. So I think the people at Chewonki were very surprised to hear that most of the area around them was closed for shellfishing. So it's a really, really pervasive problem along the coast and a really interesting one to tackle. So the model I'm going to show right now, anyway, is an FECOM, a finite volume community ocean model. It's an unstructured grid. Every one of those little triangles is a place where we solve the equations of motion and try to get its handle on circulation here. And what's cool about unstructured grids or triangles is you can get them to be really, really fine up close near the coastline, and then they get very, very big far away to sort of save you computational time. This is the Wells National Estuarine Research Reserve, but if you're familiar with the area, it includes Ogunquit. I put this one up here too because the Ogunquit River has at least 3 wastewater outfalls. We talked about mobilizing pollution, but we have a lot of wastewater treatment outfalls that happen to just go right into our coastline. So those little, I like the way they use green lightning bolts to show you wastewater treatment outfalls. So you'll notice 2 of them right here, pretty close to where people clam right here in a prohibited area, right up here as well. So you're just going to look at sea surface height for now. You see it'll start blue, and then you'll notice that as time goes on, it turns red, and you start to fill up the clam flats. So back to my theme about estuaries are annoying. They're dry half the time, otherwise why would you clam on them? So transport is really affected by that as well. So you need a model that does things like wetting and drying, that has 3-dimensionality to it, because some of this bacteria is associated with particulate matter, so it'll sink and then start going the other way on the next tidal cycle. And when you discharge at a heavy rain event discharge, if you discharge into a flooding tide or a ebbing tide, it matters. This model now includes almost 16 little streams that are potentially vectors for bringing in bacterial pollution, and we're essentially trying to provide DMR with metrics that characterize how susceptible an estuary is to a particular loading. Again, I should say that this approach is data heavy. You need at least to know something about 16 different little streams that happen within there, and that's really where Sean's team has been really awesome to work with and necessary. The other thing that's really been great is DMR's really, really hungry for getting these metrics and getting this data and using it to make better decisions. This is an example in the Harrisseekit River in Casco. They have an outfall there. You'll notice it in red. And they're using an FECOM model right now to dictate what the closure around Harrisseekit should be. So they take 1,000 to 1 dilution versus a 10,000 to 1 dilution. So this is the type of information that they're using to set closures now in the Harrisseekit River. So they're at a point right now where they can use information and say, if you told me it's not going to mix past XYZ point, we can change the map of closures. Context matters. It's the same model, except a discharge at particles at an ebb tide or versus a flood tide. So I think in the future you may see real-time calculations of where particulates might go during a particular event. This is Great Bay, New Hampshire. They have a pretty similar model to the one I'm showing. Each one of these boxes, again, is computing the circulation within Great Bay. Great Bay's got bigger problems than we do, mostly because there really are a tidal lagoonal estuary with a really long channel in order to figure out what the transport is in that area. But this is what they are using now to set their own residence time calculations. It's a 1/E folding time. So how long does it take this estuary to get to that point? And they look under flood conditions and ebb conditions, under low flow and high flow conditions. So this is the type of information we're moving towards in the Wells area. This is this annoying problem that I mentioned about transport that goes 2 ways because it's an estuary. What you're looking at now is travel time. This is work we've done in the Chesapeake to sort of give you an example. You just release a simple dye and then you figure out how long it takes to reach some other point in your estuary. So you'll notice at the surface, if you release a dye right here, there's travel times on the order of 100 to 500 days as you move around this area. If that particle is allowed to settle, then it can transport all the way up bay with the same transport. So there's a lot of subtleties and I guess annoying complexities involved in transport. But as these models become more and more common along the coast, and via CNET now we have that FECOM model I showed, pretty much along the entire coast. So those I think are going to be the important metrics moving forward in terms of residence time and how this stuff gets transported through the estuary. Is that me?
Surprise!
I put that slide in.
I could do that.
Let's actually open it up for questions in the couple of minutes that we have left. We do have some immediate next steps following up with DMR, giving a presentation to the Shellfish Advisory Council next week and continuing to coordinate amongst our team. But questions for all of us?
Thanks so much for sharing all these different amazing tools that you're putting together. I was just wondering if you could also talk a little bit about the collaborative process and how that worked, working together as a team of researchers and also working with institutions like DMR. How did the process go?
Want me to tackle that?
Could you start off?
I think what's interesting about the process more broadly is that what we've done is, I think, much more common in areas where they deal with water quality problems all the time. What we've done is tried to put together a mini Chesapeake Bay program over these course of a couple of years, where you have watershed scientists producing output that gets handed to estuarine bay hydrodynamic modelers who do water quality. So a lot of this, I think, is building the infrastructure to finally tackle this problem and say it's not okay for 185,000 acres of claimable area to be closed or prohibited all the time. So there were models, I think, to follow.
I'll also say that in terms of stakeholder engagement, some of it shows up in unexpected ways, and we had to establish in a reference watershed close connections with the community, if nothing else, just to walk across their property to set up a river gauge, and we learned a lot in the process. Also learned a lot from DMR and the healthy folks working with the Healthy Beaches programs, because they've seen different things over time in terms of where the sources are located and what bothers them in terms of gaps in knowledge and information. And so we tried to split out the problem into its source delivery and residence time parts, partly because of that issue. And then, of course, Steve Jones has been working with the Gunquit and other towns for a long time to do bacterial source tracking, and some of the information he had as well. They find some pretty amazing outcomes from their sampling, feral cats being a source, waterfowl, seagulls, culvert, the fine sediment and culverts, and so these processes are operating. But how these source delivered residence time culprits add up is pretty much undefined, but a big part of the solution. You wouldn't want to go. We also had run-ins with associations with people at different levels of government that are tasked with managing or producing responses to problems. And one of our encounters was with a local manager who was worried that we were on some kind of a campaign to actually identify coastal pollution as a problem in general without having a solution for it. And so we went down and spoke with him, not only about what we were trying to drive towards in terms of the framework and the outcomes of this project, but also the types of resources we were going to have at the end of the project that would help them in the future. And that was one of the parts they were very interested in. So that's why I separated out Toolbox from Workbench, and that you have an assembly of things now, resources to tackle localized problems.
I'd just throw 3 words on top of that. I think it's pretty mundane in the sense that it was every day. It's really routine. You have lots of meetings, emails, coordination. It was messy in that we were trying to fit together all of these diverse pieces, and it's ongoing. I mean, we really saw this as capacity building, and so we don't see this as an endpoint in any way.
So thanks so much for the presentation. And there's a tremendous amount here to dig into. And I guess my question gets at the point you just raised, Sean, which is we have a wicked problem here, and a whole lot of information was just presented about why it's even more wicked and complexified, which I love because they are, right? This is the reality, and we have to kind of go through that if we want to build better decision support tools and go after these management questions, which are fundamental. But how to do that? And you spoke a little bit to the resistance or the concern that, oh, you're just pulling back the hood and revealing how intensely complicated this management problem is, which is always a good result if we want to keep moving, beyond that. But I guess the question is really how to build some component of this that allows those solution spaces to be nearer. And obviously the work is ongoing. I very much appreciate that. I'm just curious where that is going.
But I think the more we found out, the more information we assembled, and the more we found out about these places, the more we realized. We also found out about gaps. One good example is that the polygons for the coast spatial data for the non-tidal watersheds exist. You can grab them online. There are a lot of problems with those data sets and that they have inaccuracies. But one of the biggest issues was the coastal tidal, we call them intertidal watersheds. They're outside of the non-tidal watersheds draining into the estuaries, but they surround them. Wells Estuary is a really good example. There's a big sandbar with a lot of houses on it. Those are largely not delineated or weren't delineated until we started to work on those problems. But once you have those resources at your disposal as a town manager or another type of agency, you can start addressing questions that previously were unattainable because of the lack of availability. One of the other issues is that the design of this project was that these are milestone achievements. And so one example of what can be done in the future is if you were tasked with being concerned with, or if you had a concern with runoff being the problem, not just rainfall, and you know that there are all these watersheds are unique in their own ways for source and delivery, it would be unreasonable to say you need to put a river gauge to measure flows continuously in 500 watersheds. But you might be able to do it in the 6 watersheds and transfer that data to places that are reasonable to accept the transferability of that information. And so now we have a base to do that. And so I see these as a set of milestones and actually a vision for the next strategy for the 10 to 20 year plan to get better at the work.
With that, I just want to note time. We're certainly happy to stay, I think, everybody is. But if you need to go.
This is really great. This is really interesting. It reminds me a lot of what I see going on in another collaboration, which is called ICNet, and it's bringing together climate scientists and infrastructure engineers to understand each other's models and figure out how what the climate scientists are doing can be helpful for the people who are trying to pay attention to roads and bridges in New England. They're coming up with decision support tools, and I think it would be just somehow really interesting if we could find a way to have a meeting where these two groups come together, because there's a lot of really interesting forward movement and struggles.
Do we think we have stakeholders on the call right now? Okay, so this might be unfair because it's a question I'd rather ask somebody like Cole or Carrie. But could you maybe guess what they would say based on your long experience working with them, which I really admire, about what they might feel most excited about, and maybe you've alluded to it already, but also where they would articulate a kind of frustration, which you guys have in terms of why this work hasn't been as simple as you want, and they probably have a version of that too. So this part's really working for us, and this part we still have a long way to go. You guys have something there that, in addition to what I've just heard, might help us kind of gird our loins for this kind of work going forward?
Wow. I have one example from a stakeholder that was on a conference call with Damian and me that he said on the phone, he said once we told him about the spatial data sets that were brand new, that we needed to solve some of these problems and come up with estimates, he's pretty much yelled on the phone, I want that data. I need that data. And this was a person that's in charge of managing an estuary. And or the policy implications of estuary management issues. And that's one example. The other is that the DMR has expressed interest in the fact that we've taken a diverse coastline and compiled similarities between these different areas to see that they're comparable for the same purpose that I mentioned earlier for river gauging or other analyses. So we have called that analysis, the spatial analysis, the vulnerability analysis. And what we see from that is an ability to go into places that are more vulnerable than others, which would actually be a rationale for saying we're going to invest more money in this one space because we know that the problem is elevated there, and some places on the coast have already been identified by stakeholders, partly because of the frequency and the duration of closures. But there may be other reasons that some places may just have a long-term, greater vulnerability to runoff problems and bacteria problems.
Just one quote to add to that we heard in different ways throughout the process from stakeholders, especially at DMR, that they didn't need fancy models or statistics, regression analyses. They needed management trigger levels, but we needed those more scientific approaches to be able to answer the question. So it really was this continual negotiation and working towards trying to meet their needs for those identified levels or specific recommendations about how they might change management. But it was a tension for sure.
In psychology, we say, you got the type 1 and type 2 errors going on here. Is it really more dangerous to miss something that could happen, or to set the bar so high that it's has economic cost? And are there economists or psychologists involved in your team thinking about those kinds of issues and how that impacts the management sort of perspective? Not that there needs to be, but
well, there's a lot of work on that, right? Because if you set your overly liberal, so to speak, and people get GI problems all over the state, it has this halo effect that reduces shellfish consumption. So I think they are sensitive to that for sure.
there are lots of interesting questions there about which things do people hang on to in terms of their worries. If somebody who has high visibility, gets sick, does that make more of a difference in sort of how to understand when people pay attention to the kinds of things? I mean, if we all get sick as academics,
Well, I mean, the best situation would be is if you had real-time monitoring in all places at all times, and that's unreasonable. But the interesting part of this is that there's a freshwater flow going into a tidal estuary, and that those mixing dynamics are part of the issue. But there's these random processes, these processes that are occurring randomly. The sources, in particular, are a big part of that, or potentially a big part of the story in some places. And so, what that means to me is that people may have a sense for decadal timescale management approaches. Say you're going to sample repetitively in the same spot, but it may not be good enough. You may need to sample in other places. We've uncovered several issues with the framework for sampling that were relevant to our statistical analysis and created biases. So expanding out the data set depending on what the question is. Some places that were thought of as having a low risk weren't sampled as heavily as places that have a high risk. But if you're parsing out the state into vulnerability zones, that matters to the outcome. So how to track with police records or maintenance records or so forth, and how that could feed into the DMR decision process is one of the things that has come up. The role of large rivers. The large rivers were not in our data set because they're too big, the Penobscot, the Kennebec, and there's others. But they may have an impact on it may be a regional problem and not a local problem in some instances. So I always think of that, what do they say, think globally, act locally. But maybe in some cases it's think locally, act regionally. Maybe large river inputs matter to other places on the coast. And I think we have a step in the direction of seeing that some of those gaps exist. We need to.
The last really interesting idea I think that came at the end of this is thinking about success. So if there were no closures on the coast, for instance, the management of the resource would then fall to these local municipalities, because this is a locally managed species, which is very unique to Maine in many ways. But if you have 11 closures, a year, or 9 closures a year, essentially that's also time for clams to grow without being harvested. So I'm really fascinated about success and what it looks like. Because right now we have lobster that water quality doesn't seem to matter really. It's nothing to do with it, right? And then we have the second largest fishery in the state is centrally governed because of water quality, basically.
And just going back to your original question, so half the team were social scientists, including behavioral economists and economists. But interestingly, as I watched this part of the process, some of the lead stakeholders initially wanted more from the biophysical side, and so all of these good folks worked to deliver it. But over time, I think they've come to appreciate much more about the human dimensions. And I will say that some of those talks have been given here and more will be, but that's no less complicated than this, not the least that people who, when they get out to a beach where there may be an advisory posted, don't even look at it, or look at it and come to a totally different conclusion than whoever wrote it about what it means in terms of risk. So bringing all that together is something that's a challenge for us and apparently everybody else who's trying to make sense.
I think that's one of the things that makes this particular problem interesting / challenging. Is right. I mean, you expect the social side of a coupled natural human system to be complicated. It always is because people are complicated. I'm not saying the science is easy, but sometimes the science is established and straightforward. But you know, as we were maybe at the take-home message across all of our talks, is the science is so complicated on this side. It's so messy on this side too, and so you know, it's a wicked squared problem.
Very nice.
Paper title.
Your figure does that the best. I mean, it really demonstrates that there are winners and losers. If you have a negative relationship between a 2-inch rainstorms and bacteria, then you're a loser in these closures, right? It has nothing to do with you. So.
Go ahead. Another exciting part of this that we've talked with the stakeholders about is that one issue is can we have a better set of rules? The other is how do we go forward down the road, and there's climate change and land use change in different places. And so knowing the source and delivery problems, if they exist, are potentially manageable. You may not want to have a high-level development in a place where you have a vulnerable clamshell clamming flat. That may be one issue. You may put in stormwater management of a certain type in an area to control runoff rates, and you may not do it in other places and spend as much money on it. So the allocation of funds. But the other thing that's interesting is the source and delivery problem from the watershed is different than the residence time problem. You may have a very hard time managing a residence time. I think of, what was it, Ross Perot blew up a reef off of Bermuda to get his boat in, but I don't think that's going to happen. And these issues are hydrodynamics of a local area are at the mercy of the geometry of the area. And Sam spent a lot of time with these dynamics, and some places have low flushing times, and so they're potentially more vulnerable, and that needs to be known when you look at the changes down the road and how they affect the relation between source delivery and residence time.
So clearly we could keep talking about this for the next couple hours. We might need to go somewhere, but thank you for staying and for joining us.